pleasure to introduce Dr. Kouokam. He's an assistant professor in the Pharmacology and Toxicology Department, and his research includes the development of vaccines. We collaborate on some projects to inhibit HSV2 and HIV and other bacterial infections, and then he also works on projects developing anti-cancer and anti-HIV drugs. So a little bit of history about Dr. Kouokam is that he entered graduate school, or that he was awarded his BS in biochemistry from the University of Yaoundé in Cameroon. He then entered graduate school at the same university where he received a Master's of Science in 1995 and was admitted for Ph.D. studies. Subsequently, he was granted a German scholarship, the DAAD Award, to pursue his graduate research at the University of Saarland in Germany, and it's there that he earned his Ph.D. in 2002 with a focus in analytical phytochemistry and pharmacognosy. So thereafter, Dr. Kouokam moved to Sweden for a postdoc in molecular pathogenesis of uropathogenic E. coli, and then back to the US for a postdoc in microbiology and immunology and in pharmacology and toxicology at the University of Louisville. And as I mentioned, he's currently an assistant professor, and I've enjoyed collaborating with him, and it's an honor to have him present here to you today.
Thank you, Jill, for this warm presentation, and thank you all for coming. I hope that you're not going to be disappointed at the end of my talk, but I will do my best so that you are convinced that lectins are very important in life and can be used as therapeutics. So you have seen my title, which is plant lectins as multifaceted drugs. I will present the case of Griffithsin, not because Griffithsin has a problem, but because Griffithsin is a very good lectin. So my talk will basically start with an introduction, then I will, it's a long introduction, I just warn you, and then I will move on to describe a few lectins for viral and sexually transmitted infections before presenting a little case why this protein can be used as anti-cancer agents. Then I will introduce Griffithsin, a few future plans, and then conclude. So what are lectins? This is a question that everybody should probably be asking if you don't know it already, or at least if you know a little bit, you may want to know more. So this started in 19th century by a man called Stillmark, and he wrote a doctoral thesis that was entitled Über Resin ein giftiges Ferment aus den Samen von Ricinus communis und einigen anderen Microbiciden. This is a German title, meaning just that he is writing about ricin, which is a toxic protein from castor beans, which is also called Ricinus communis. So ricin is very known by many of us, and not for a good thing, but I will talk about that later. More proteins have been isolated from plants and others with similar properties, and you can see some of the names here: phytoagglutinins, phytoimagglutinin, and so on. And in 1954, the term lectin was actually coined by Vogt and collaborators, which actually means it stems from the Greek, from Latin of pick out. So basically, lectins can pick out some stuff from a meat. I will explain that later, hopefully. Then the definition was extended to include all the protein of non-immune origin that can agglutinate red blood cells and other cell types. So lectins are very kind of broad, are very ubiquitous proteins, which are nowadays defined just as carbohydrate binding proteins. So everything that is carbohydrate binding proteins can be called lectins by now, and some of them have enzymatic activity, some don't, but they are all have this property of binding. They all have this property of binding proteins. So these are the potential uses of lectins, and I will go one by one through this in the next few slides. So blood typing, It's a rather complex topic, but to simplify the thing, I will just display the most common blood grouping system that you have here, ABO system, because there are 35 other systems that are recognized. So, but in this system, you have the people of the A group have antigen A. I don't know if you're going to be able to see it, because I have probably no pointer. Somebody has a pointer? No. Okay, thank you, sir. All right. So, group A, people have antigen A on the surface of the red blood cells, which means in the plasma you're going to find an anti-B antibodies, and in group B is the opposite. You have the B antigen on the surface of the red blood cells, and you find the anti-antibodies. In group in AB individuals, there's no antibodies because they express both of these antigens on the cell surface. Now, group O have no antigen at all on the surface, which means they can basically they have both anti-A and B antibodies in their plasma, and this determines how blood transfusion can be made without risk, because in the past people died a lot from blood transfusion. And so what do lectins have to do with this? Lectins can bind preferentially to these are different lectins, and you can see the blood group here. Some are very specific to certain blood group like HPA, and even some that will be slightly different because they are 2A. They are A1 and A2. A2 just means that it has less specificity than A1. So they are the same, they have the same antigen, but A2 is a little less specific. And on this list here, you have things that you know very much of. A soybean. This is peanut, and which led some people to actually think some people should worry about lectins, because if you, these are stuff that can agglutinate your red blood cells and cause a lot of trouble. And the concept of blood group diet was proposed, meaning that you should be careful of what you eat according to your blood group. And it's not very surprising that at some point you may see something like this, caution lectins, because this might be the next gluten we are talking about here. And I had this passage here that everybody can read from a review online that basically think that people should be wary, not worried, but wary, which is almost the same in some sense, about lectins, and that they should, and these are found almost everywhere. You can see the list, it's almost everywhere you can find them. So, but lectins can also be used as a tracer agent for neurons. So they can inject lectins in the brain, and that allow the visualization of the neuron and the neuronal networks. In this case, PHA, phytohemagglutinin A, is the most commonly used. I put concanavalin A from Jacobin side by side so that you can understand how the structural differences can actually make a huge deal in terms of use of lectins, because lectins are determined by their binding specificity, which is dictated by the primary structure. So the sugar that they're going to bind depends on the structure of the amino acid they have in those pockets. And the oligomerization is very important too, because if a lectin is dimeric, that means it has twice as binding site as the monomeric form. So it's very important as well. And the conformation, you see here, both are tetrameric proteins. So here you have two dimers that are parallel to each other, and in concanavalin A, you have one dimer that is basically perpendicular to the other one, to the other dimer. And this is very important to the activity. This is how they do the tracing with phyto A. Sorry. I'm not going to go into detail, but just to show you that whenever they inject that in brain, they can actually see a lot of structure from brain cells. So one of my things that is close to me is as an analytical chemist is affinity chromatography. So lectins can be used in affinity chromatography just by coating the lectin on a resin and loading in the column. Now when you bring a bunch of protein up there, only those that can bind to the resin, those are the glycoproteins, will be able to stay at this point here when the other ones are eluted. And in the next step now you can just remove your protein of interest, which is very cool and very simple to do. Now, as I mentioned, even as Denmark discovered in the 19th century, lectins are not only for a good cause, they also can cause harm. And in this case here, I'm presenting the some picture of ricin. This is castor bean, and it's very easy to make ricin because it's basically the leftover after you make the castor oil. So you note all the threads. The next slide will show you a few events that happened in the history. So this is a dimeric protein. Ricin is a dimeric protein. You have the red subunit, which is the A subunit that's actually active, and the B subunit that is a lectin, which is which help the lectin to bind to the surface of the cell. So when the lectin binds to the surface of the cell, it may be on glycoproteins or glycolipids, because it just needs sugar, basically. It now is internalized in the cell. Now it travels the cell to the early endosomes, where it moves to the Golgi apparatus before entering the ER. In the ER, there's an enzymatic cleavage of the A subunit that actually goes backward to the cytoplasm and bind to the ribosome the 28S subunit of ribosomal RNA. So now the protein synthesis is stopped and the cell has to die. So that's how ricin actually causes a lot of trouble. We talk more about ricin, but heparin is also very toxic. Actually, it's more than about 30 times more toxic than ricin. And this can be inhaled or injected as well as ingested. Here is mainly IV route that they have described. But they have the same mechanism. So these are a few events that you may recognize some. Even our former President Barack Obama was once received some letters, with ricin, and this has been going on since about the time I was born. So it's a long time. So now, what do you believe lectins could be used for cancer treatment? This is because not only you have a few headlines in the literature showing a few lectins that actually have some anti-cancer properties, but we also know that the lectins have on the cancer cells have on their cells immature glycans. So since they divide so fast, maybe they don't have the time to to complete the maturation of the glycans as the normal cells. So what happens is that these glycans are very prominent on those cells, meaning that you can specifically target cancer cells as opposed to normal cells. But of course, it's a little bit challenging because that's not as straightforward as I'm saying. But there's a possibility there that you can use lectins to target cancer cells. This is probably where a field where lectins are being used a lot, because a lot of lectins are being assessed for their antiviral activity against enveloviruses. Just to name a few enveloviruses that have been described as susceptible to lectins, you have flu, you have HSV to the both type 1 and 2, you have coronaviruses, and HCV, even HBV has been discovered in some instances, and of course HIV. So I will try to describe what happened in the case of HIV. So in this cartoon, you see the life cycle of the HIV. So what happens is that it starts with the binding to the cell surface, primarily on the receptor called CD4, but this needs the help of a co-receptor called CCR5, but it could also be CXCR4 because not all viruses express both. And once that is done, the virus will attach and get into the cell cytoplasm, where it allocates where there's a reverse transcription here to do the cDNA, and then the cDNA now goes into the nucleus for replication and all the subsequent processes before being exported to the cytoplasm again to translate the viral proteins, and then the viral particle will assemble again and come back and start the cycle all over. Nowadays, we know that many antiviral drugs have been targeting specific steps on the cell cycle, on this viral cycle. Lectins can specifically bind to the sugars here, so preventing them to be able to attach or to bind to the CD4 cell or the receptors. So they can also intervene at this stage of attachment. That was has been shown in the literature. So in the case of now we want to see if Griffithsin can also help to target or to manage other STDs in addition to HIV. So I just wanted to have a snapshot of what actually this means in terms of disease burden and also in terms of cost. As you see here, if you believe it or not, HPV, human papillomavirus, is the #1 STD in the US. This is about 14% of the population. And there are others that are here. If you look at HIV, it's actually one of the least in the US, and basically it's more prominent in the US now among MSM. But in terms of cost, HIV actually takes over everybody else, with $12.6 billion of dollars every year. So that's a lot of money there. But STDs are very important in microbicide development, because whether you have STD that cause inflammation or ulcers, you're going to end up increasing the susceptibility to HIV. And in the same way, the STD will increase the virus shedding, which also increase the infectiousness. So what happens is that STD means more HIV. And in turn now, HIV will decrease your immune system, so that will increase also your susceptibility to other STDs and also the severity of those diseases. In the meantime, your drug will be less efficacious because of poor immune system. So that means you have a synergy, a biological synergy between HIV and STDs, which actually amplify the whole thing. And there are still questions that are raised to know if this whole thing actually are important in what they call HIV super infection, which just means that an individual that's already infected with HIV have a different strain of HIV again, because you don't have to have only one strain of HIV, you can have 5 or 10, whatever. And that, in that case, they call super infection. So we are interested in a few pathogens, sexually transmitted pathogens. including Candida albicans, Chlamydia trachomatis, and Trichomonas vaginalis, which are very important in terms of disease burden. You can see the incidence here. It's very huge, and the odd ratio to have HIV when you are carrying those bugs is very high as well. So in addition to this, we also want to see what happens with UPEC, which is not theoretically a STD, but research has shown that sexual partners tend to have the same strain of UPEC, meaning that maybe it's transmitted sexually as well. And a lot of people have UPEC, so it's something that it could also be interesting. And the most interesting thing is that all these pathogens have on their surface some sugar proteins, some sugar binding, some sugar containing proteins. They have different names, but they're all sugars, and all sugars can be bound by lectins. So now I'm moving slowly to Griffithsin, which is actually what we spend most of our time working on. But I'm not going to talk that much about production, because production can be another topic. If you are interested of my talk, you can call me another time. I will mainly briefly try to convince you that Griffithsin is a very potent antiviral, but also that it is very safe. And I will also give you some of the problems we had with Griffithsin and how we try to solve. So this is the ID card of Griffithsin, which is of course a lectin, and it was originally obtained from a red algae called Griffithsia, and this was off coast of New Zealand. So it belongs to what is called jacqueline related lectins, and it mainly binds mannose and also glucose and N-acetyl glucosamine, and also other like salicylic acid and others. So it binds a lot of sugars actually, but the main or the most prominent sugar that is specific is mannose. Now, it's usually found in dimer. Actually, there are questions about whether you can actually find a monomer, a Griffithsin monomer. And it has broad antiviral activity against viruses. And how it acts is by tightly binds high mannose containing glycoprotein on the viral envelope. And the structure is just a small peptide, 121 amino acid, with the residue at position 31 replaced by alanine, because that was a non-common amino acid. Nobody has ever found the structure, but it was replaced by alanine just to synthesize the protein in plant or bacteria. And it's a beta phase 1 structure that's very common to this JLR. But the specificity of Griffithsin is that the two monomers must actually share some sequences, because if you look carefully, the blue is one monomer and the green is another one, and you see that the blue contributes to the green and vice versa. So it's they call domain swapped dimer. So I already say what happened with Griffithsin. It actually binds this sugar. If you look at all these enveloviruses I've placed before, you're going to see like coronaviruses have the spike proteins, spike glycoproteins here, which of course have sugars. And the HIV has GP120, which primarily bind to Griffithsin, but you also have the GP41 that are all glycosylated. So Griffithsin is the most potent anti-HIV lectin discovered so far. And this is a list of other lectin candidates that have been assessed for their anti-HIV activity. As you can see, Griffithsin is active in the picomolar range, 43 picomolar. So the closest comes like 3 times less efficacious. But the problem is that this CVN, which was for a long time a very a very highly regarded lectin, had a lot of toxic activity, so they kind of drop it. But Griffithsin is not only active against HIV, it's also against a lot of other viruses. This shows you the activity against HCV. I will not spend too much time on this, but I just want you to see that if you treat HCV, this is a control group, you treat HCV with Griffithsin, you're going to end up having complete inhibition. And they have a mutant of Griffithsin that doesn't bind sugar. So that one doesn't have any effect at all, meaning that the activity you see here is actually lectin related. And they also assess the cell-to-cell transmission. So what they do is that they take they infect cells, and then they co-culture cells with those that are not infected, and see how the infected cell will spread the virus to the next. What they do to prevent cell-free transmission is that they will treat they have a control here that is anti-HIV, HCV antibodies. So they're going to bound all the free virus in the medium and allow only the virus from the cell to actually infect the next cells. So if you see pink here, the pink nucleus means this is the donor cell. So this is the cell that was originally infected. And the one that here have a blue nucleus means that they have just acquired infection. So as you can see here, with the antibody, you still see this infection around. But when you treat with Griffithsin, you only see the pink nucleus and green cytoplasm, meaning that there was no cell-to-cell transmission. So I stopped the cell-to-cell transmission. So next, we also, they show that Griffithsin inhibit coronavirus both in vivo and in vitro. So this is just how it does in a dose-dependent manner in vitro. And in mice, as you can see here, Griffithsin and SARS protect completely mice against SARS, while the sham group, is while the SARS group only is completely, or at least 75% are gone at some point. And that was also the case for JAV and in vitro and in vivo. So I'm not going to spend too much time on this. HSV2 is also an interesting case, and in this paper, what they did was to treat mice intravaginally with Depo-Provera treated mice. So they will inject a single dose of Griffithsin to Depo-Provera treated mice, and then they will challenge those mice with the virus. So 15 minutes later, they will image 1 and 4 days after infection, and then this is what they see. So this is how the mouse is supposed to look like. So this is a mouse that has received nothing. Now, if you have treated Griffithsin, you barely see anything as well. This is the first day. And then in the carbopol, which is a gel, a control gel, you see that a few mice are already showing a huge signal. And after 4 days, although you have one mouse here that starts to show signal, most of them showed no signal, and you still have the infection in all other mice. So this is a very impressive finding, because it means that Griffithsin is has protected these mice from infection with HSV2. And in our lab, we have actually demonstrated that this can go up to days, at least 3 days. So at least you can treat the mice with Griffithsin and wait for 3 days, and you challenge and still have the protection, which means it's actually it's very protective against these viruses. So I did some experiment to see whether this will apply to Candida albicans, and I first started with seeing how Griffithsin will affect the sugar, because Candida albicans has some sugar on the surface called mannan, and I just took those purified mannan and treated with Griffithsin, and as you can see, there's a dose-dependent. I could still show the binding, and I could also show the inhibition of the signal from mannan if you treat with Griffithsin. And I used GFP conjugated Griffithsin to culture or to treat Candida albicans, and you can see that the surface was all covered. And this one just was not with the unlabeled or with the mutant Griffithsin that doesn't bind the sugars, meaning that this what we see here is actually lectin related as well. And it also inhibits the growth of Candida albicans to some extent. I mean, although the inhibition was not complete, but you could see that there's a dose-dependent inhibition of Candida albicans. So we concluded here that Griffithsin binds and inhibits C. albicans, and by binding the surface mannan, and also that GP120 binding of Griffithsin is inhibited by the SC mannan. So this is actually very promising. We are planning to write some grant proposal with this, and hopefully get funded. So not only that, and very surprisingly, Griffithsin also inhibited HPV. HPV is not an envelovirus, because I've been saying that enveloviruses are susceptible, but HPV is not one. But what happened is that when they combine Griffithsin and carrageenan, they found a very powerful inhibition. And the problem here is that Griffithsin doesn't do anything on the envelope, of course, because there's no sugar. But it can bind to alpha-6 integrin that actually help the virus to spread in the cytoplasm. So by blocking that step, you also have reduced viral load. So Griffithsin is usually thought of as a microbicide, meaning that they're going to have mucosal administration. But we were interested in knowing whether Griffithsin can actually be used systemically, because just imagine XCV, even XRV to some extent, all of those can be found in the bloodstream. So how do you actually go there and get the Griffithsin? Is it going to be safe? Because it's a lectin, so you don't know it's going to agglutinate, whatever. You don't know what's going to happen. So we treated mice. First of all, we treated mice with Griffithsin and single dose, very high dose, and also 10 daily dose of a lower dose. So this is a chronic treatment, that's an acute treatment. And what we found was that actually you have Griffithsin even up to 15 days here after single treatment. You still have enough Griffithsin here to inhibit XRV. And in a daily treatment, 10 daily treatment, what we found is that after a certain point, after probably 8 to 9 days, it doesn't matter if you continue to treat, the Griffithsin already saturated. So what happens is that it starts decreasing, but you still get some there at around 21 days, which means 11 days after you stop treating. And so this was very interesting, and that was also repeated in guinea pigs. So we concluded that Griffithsin contains serum sample. Okay, we found that this Griffithsin sample, containing serum sample that we had from this animal actually had anti-XRV activity. And then it was also well correlated. I will show some data about the safety later. So we said Griffithsin may be developed for the treatment of systemic infection by susceptible envelope viruses and help eradicate HIV. So we went ahead and did the pharmacokinetics of Griffithsin, and we found that it actually has a very high half-life, about 12 or so hours, which is good for a therapeutic. So what happened is that when we treat just once, just a bolus dose of Griffithsin at time 0, you can still detect at 72 hours and even 96 hours. So that's 4 days, and subcutaneously or even intravenously. So, which means that Griffithsin can actually be used for systemic treatment with a good efficacy. But not only that, we found out that when you treat Griffithsin systemically, when you inject subcutaneously, you find Griffithsin in virtually all fluids. You find in urine, and actually that's where you have the most. You find in fecal extract, you find in vaginal washes. We didn't check rectal washes at this place because we were more interested in vaginal microbicide at the point. We find in nasal washes and so on. So you find almost everywhere, which means you can target those places with Griffithsin just by administering systemically. So now, with this great efficacy, we wanted to know the safety property of Griffithsin. And so this is basically when I joined the lab in 2008, I was presented as the task to try to sort out how safe this protein might be. So we first started with looking at explant. So this is ectocervical explant from a young woman. And this is just to show you the general structure. What I'm going to show here is just that when you treat with labeled Griffithsin, you end up having all the outer, the outer surface of the ectocervix that is covered by Griffithsin, and the mutant doesn't do that. This is the mutant that doesn't have a lectin activity. But when you treat with CBN, you actually have a whole different pattern of penetration of CBN, which actually is not very good, because this might have some off-target activity that you don't know. So we did the same for human PBMCs, and you can see that Griffithsin. So the green here show you. So if there's banding, there's going to be a shift to the right for the green. So you have Griffithsin that will shift the thing to the right. But if you treat with mutant, you have no shift. And with CBN, it's a very complex figure that we see here. Just showing you that Griffithsin is actually binding to the cells. But what happens here is that if you treat PBMC with Griffithsin for 24 hours and wash them and infected for 9 days, Griffithsin is still active. So which means actually, this binding is, is there for a long time. So, which is also intriguing, because you don't know if binding will cause some harm or not. Because in the microbicide fight, the golden rule is do no harm. So you cannot try to sell any microbicide that is doing even the least harm, because nobody's going to buy it. So we then did microarray experiment just to see if there's any effect of Griffithsin on gene expression. So this is just a heat map showing you Griffithsin at different concentrations, and to compare with the PBS here. What you see here is that at this concentration, Griffithsin is actually very close to the PBS. But there's some toxicity when you increase the concentration, but remember that it's active as picomolar range. So meaning that this is very high, these are very high concentration to start with. But in the case of CVN, you see that everything is completely different from the PBS here. So when we did some basic quantification here, we realized that Griffithsin affect about, I can't read very well, but 100, let's say 100, a few hundred genes here at 1 micromolar. And just for your comparison, the similar plasma, when you use for the same cell, affect more than 400 genes. So meaning that Griffithsin actually safer than the semen in that case. And so we went ahead and did the same experiment in the mouse model because we wanted to show, to do more experiment in mice. So we wanted to validate what we found in the mouse model as well. So here, just showing you that when you treat mouse PBMCs with Griffithsin, you have no release of cytokines, or you have no effect on cell viability, or even mitogenicity of PBMCs. So, which was encouraging. Then we actually did the safety studies in the mouse model. So here just show you very rough, and so this is just body weight, and then you have here what is this? I can't read. Okay, so this animal weight, sorry, this animal weight here, and then you have here the what? Okay, yeah, right. So here is animal body weight, all of it. And these are different treatments. So this is higher dose of Griffithsin, and this is the chronic dose of Griffithsin. So when they have the same weight, I mean, there's really no change here. When you go to spleen body weight, so we are interested in spleen, we actually check all the organs. But the prayer, we realized that there was some increase in spleen weight, as you can see here. So we decided to just display the spleen result. So you see that there's some splenomegaly that is, visible in almost all of this. But the good thing is that it is reversible, because when you treat with Griffithsin, after some time of recovery, the spleen actually goes back by itself. So that, which means is reversible. And you find the same with the chronic treatment. So it's not something that is very it's very common with drugs, to increase the size of the spleen by a little bit. But of course, if it is something serious, we're going to be able to see it on histopathology. And we didn't really see anything that much on histopathology, but we are interested in knowing if actually the splenocytes are activated in that case, or they are just increased in number, or if they are activated. Because if they are activated, they might induce some immune system, and we don't want to do that in the context of HIV, because more immune system you have, more susceptibility you actually also generate. So, but overall the B cells were not very affected, and the T cells were not activated at all. So meaning that this is something that is not of great relevance for the use of Griffithsin. And we now try different routes of administration. So we try intravaginal, intraperitoneal, and subcutaneous, and also found no real difference whether in body weight or spleen weight and so on. And here we use a slightly lower dose. We use a dose of 2 milligrams per kilogram of Griffithsin here instead of 10 before. And even this is still a lot of Griffithsin. I mean, we can only use this in mice. You cannot try this, in larger animals. So it's still a lot, and we didn't see any consistent toxicity. So in summary for Griffithsin, I hope you agree with me that it is the most potent anti-HIV lectin described so far, and has an excellent in vitro safety profile, and also in vivo. And it remains very active after binding to cells. And you can also get in mucosal fluid after you treat systemically. So this means you actually have several other potential uses of this lectin. So then came the problem. because we were so excited until we started formulating Griffithsin for use because we are running a macaque study in Atlanta. So we were so excited, and then our collaborator realized that Griffithsin was actually susceptible to oxidation. So with time, we were losing some amount of Griffithsin, not too much, maybe 4% or so, but it's enough for FDA to give you all the trouble to try to characterize the 4% and try to show that the 4% doesn't affect activity and so on. So our group decided to instead replace a methionine at position 78 by an alanine. So by doing that, you actually prevent oxidation, and that has been shown over and over that this is very similar to the original Griffithsin, and that is a better candidate for microbicide. So, but again, our job was to show now that this is safe, and we went back to cells. This time we used colorectal cells, we used Keiko2 cells, colon cancer cells, and also some other cell lines, and we showed that there was really no effect of Griffithsin on viability. But if you use con A, which is another lectin, we're going to see a very big drop of viability. So this was the same when you treat PBMCs, human PBMCs with Griffithsin. There was no effect on viability from Griffithsin or mitogenicity. So meaning that we could, it's similar to the wild-type Griffithsin in terms of safety in this case. So the same was observed when you look at many cytokines. So in this experiment, you have 5 different donors, 5 PBMCs for 5 different donors, and each donor we assess a few 26 or 7 cytokines. So what you see here, so it's from the complete, so it goes from brighter to darker, and then what you have is, whenever you have something that there's no effect at all, and this dark means there's a lot of effect. So this is just for comparison. What you have with con A at 1 micromolar, and this Griffithsin at 4 micromolar and at 1 micromolar. Here you barely see anything, except for this patient who was actually showing some higher level, increased level everywhere. There was barely anything to be seen. Maybe the patient has some other underlying pathology, we don't know, but anyway, in general, it's very good looking profile. In microarrays, there were some genes that were changed, for sure. But what we know is that if you treat Griffithsin with these Keiko2 cells, you end up having Griffithsin and PBS that will cluster together, while the con A is completely out. So which means that although it's affecting a few pathways here, it's relatively safer than con A. But this is a cancer cell line, and I checked those pathways. Most of them were actually cancer related, which is not going to happen, in normal situation. This was maybe it was just something. So we don't know if it's a good thing or a bad thing that is affecting this pathway anyway. So I'm very close to the end. So I'm going to say a few words about what is coming up. The first thing is to further develop Griffithsin as a microbicide. And we will, in collaboration with Jill, we are planning to test Griffithsin formulations against STDs, this one here, and maybe others. And also, we are interested in looking at efficacy of safety of Griffithsin in the context of colorectal pathologies. So in this case, we treat mice with DSS, and we see whether Griffithsin is still active against HLV2, or if it's still safe in that context. So that's what we plan to do next. And as I was saying, we are very interested in plant lectins for cancer activity. And this, we are planning to use lectins as a platform to start this project. So I'm sure all of you know about mistletoe, hopefully, but somebody asked if it's only for, if it's just for Christmas or also for cancer. And this question is not naive. It's because in Europe, especially German-speaking countries, mistletoe extract are very used as therapeutics against cancer. So this is not a naive statement. But in the US, it's not approved, and this is because the FDA didn't like the idea of extract. First of all, extracts are very variable. There's no one mistletoe. There are 1,000 mistletoe because every tree has a mistletoe that is different from the other tree. So when you see 1,000 mistletoe extracts out there, they are all different. None is the same. So the FDA doesn't like the idea at all. So that's why we are trying to go to the lectin level and see if we can actually get something out of it. The active molecules that are known in mistletoe are lectins. They are viscotoxins and flavonoids, and also membrane lipids. But we are more interested in mistletoe because there are some indication that it might be actually active against cancer. So, and yesterday I saw that there's a GoFundMe online on cancer, on the use of extract, mistletoe extract to treat cancer, a clinical trial in the US. I mean, I'm not encouraging you to, I'm just showing you information, so I'm not part of it. So, but as I was saying, even that we have trouble with FDA because they like single molecule and they like to characterize things that, that can be traceable, which is not the case for mistletoe lectins. Even the same plant, different seasons produce different extract. So it's very important to go to the lectin and see what happens. And you can also try to mix, to mix the lectins according to the ratio that is found in plant, because we know that ratio. We know those are active, like you can try to do that. And the advantage we have is that you can actually produce them in tobacco plants. You can produce protein in tobacco plants. That's what our unit does. So if we do that and we have pure mistletoe lectin, there are 3 of them, and we produce viscotoxin as well, we can mix, or we can just try single one and see what happens, and in terms of activity or safety. So it's a lot said, but I'm going to summarize in a few words. Lectins exert their effect by binding to sugar moieties generally found on cell surface, and they have great potential for use in the treatment of envelovirus-caused disease, and also they can be used against other STDs, and of course they are potential target for cancer cells. So now we strongly encourage that some other study be carried out to try to sort out these therapeutic properties. And with that, I'm done. But I come to my favorite slide. This is where I can spend the next hour if you allow me time. But I'm sure you won't allow me time. So I will just try to be as brief as I can. So I work in the biopharmaceutical research unit that's headed by Dr. Kenneth Palmer, who is my boss, but a lot of other things. He's not only my boss, also kind of friend, sometimes kind of brother, sometimes, because he comes from Africa, by the way. So a lot of things. In that group, you also have Nobi Matoba, who is a PI, and George Fukwe, Dr. Fukwe. And there are a lot of people. I just mentioned here those that I have closer relation with daily, including Sanet, Dr. Nabeta, Amanda, Joshua, and Dr. Hamoski. And my collaborators, I want to thank Jill Jill is a very dynamic person, and actually it's because of her that I'm standing here now. And I want to thank you for the opportunity also to, to bring me here to see new faces, and hopefully they see mine. And I hope we're going to have a long time together as collaborators. And I would also to thank my friend Josh Wood in the Department of Pharmacology. This is my, when things are good we talk, when things are bad we talk, and so he's a good friend of mine. And also Lisa Sander that that want to help us in some part of our grant that we submitted recently. Of course, I was saying I could also put this name down here, but I don't want to repeat names. For animal work, Sarah was very helpful when I was doing animal work, and Dr. Bauer as well as Ashley. In Farm Talks, all the faculties are nice to me, but the head is nicer than everybody else to me. So I want to acknowledge that. And I was appointed as a faculty in the CPM last June, and I want to acknowledge all the faculties there and all the staff. The funding that currently we have is from the supplement grant to Kenneth's U19 grant, and also a CPM startup that we have. And of course, there's nothing like friends and family. And I have five kids, and a beautiful wife. With that, I want to thank everybody for coming and inviting me for your lunch. Thank you so much.